Hello, and a very warm welcome to today's Select Science webinar. My name's Charlie Carter. I'll be moderating this presentation, and I'm delighted to be joined by Patrick Brown, who has a fantastic talk lined up for us today titled mRNA Vaccines, Development, Manufacturing, and How NanoDrop Eight Can Help. Please submit your questions for the Q&A session at any time during the webinar using the button to the left of your screen. And without further delay, I'd like to hand over to Patrick.
Right, well, thank you for joining me today. My name is Patrick Brown, and we're going to talk a little bit about mRNA vaccines, their development, manufacturing, and how our new Thermo Scientific NanoDrop Eight microvolume spectrophotometer can help in that workflow. Now, we don't have a whole lot of time, but we're going to go through these 5 agenda topics. So we'll go through a brief history of vaccine development and the background for that topic, mRNA vaccine methodologies and research areas, and mRNA vaccine production. Now, each of those 3 topics could be a webinar or an entire webinar series in and of itself, so we'll touch on all of those at a high level. Then we'll get into UV-Vis spectroscopy and why it's a useful tool for quality assurance and quality control in this whole mRNA vaccine world. And then finally, we'll wrap up by talking about how the new NanoDrop 8 instrument, which is our latest 8-channel instrument, can be used to aid in that whole process. So we'll get started by talking about a brief history of vaccines and some of the vaccines being used today. Really, vaccine research started in the late 18th century with Edward Jenner's work to make a vaccine against smallpox. About 100 years later, Louis Pasteur's work centered around the isolation of the pathogen, then inactivation, and finally injection. In its simplest form, vaccines are presenting our body with inactivated pathogens or part of the pathogen to train our immune system so that when it is presented with the pathogen in the future, our bodies will be able to fight off the infection. In the 1950s, the foundations of traditional vaccine platforms that we're familiar with today were formed. And this was really enabled by the innovations in propagation of virus in cell culture. The traditional methods that we're talking about are kind of shown in that top section there. So the traditional viruses, the virus-based vaccines, the traditional vaccine platforms are broken into virus-based vaccines and protein-based vaccines. Kind of in that top left-hand corner, virus-based vaccines can be then subcategorized into 2 different categories. So inactivated virus, and that's where the pathogenic virus is propagated in cell culture, isolated, and then heat inactivated. And one of the examples here is the polio vaccine, which saw large-scale production and vaccinations beginning around the mid-1950s. The second category of virus-based vaccines are the use of live attenuated vaccines. Now, this platform's steps involve isolating the pathogenic virus and then passaging the virus multiple times through cell lines. And it's during this multiple passage process, the virus accumulates mutations that reduce its virulence. The MMR vaccine and some of the seasonal flu vaccines are examples of vaccines from this category. In the top right-hand corner there, protein-based vaccines are the next major traditional platform, and these involve the use of virus-like particles or individual proteins or protein subunits of the pathogen, and the HPV vaccine is one of the examples there. Moving to the bottom, we get into the more modern or so-called next generation vaccine platforms. On the bottom left, nucleic acid therapies have been a very popular approach to creating new medicines since the 1990s, when advances in the genomics field enabled innovation in this arena. At this time, the use of nucleic acids as vaccines began to be investigated. And initially, much of the early work was being done using DNA because it was much more stable than RNA. So plasmid vectors expressing the antigen of interest would be introduced into cells where they would be transcribed and translated into the protein antigens. And DNA delivery into cells within the body became a large barrier to overcome. In vitro, electroporation was the most efficient option. However, this is really currently not an efficient option in vivo. Initially, RNA represented several challenges. Most notably was its instability. And later, I'll talk about several of the innovations that took place about 10 years ago, which has led this to be a viable vaccine platform today. The final category is the use of replicating or non-replicating viral vectors. And this platform involves cloning antigen genes into a well-characterized viral vector like adenovirus. Again, one of the examples that we'll talk about or that's given here is the Ebola vaccine. So now let's talk a little bit more specifically about mRNA vaccines. The first mRNA vaccine methodology that I would like to describe is the use of non-replicating mRNA. Here in this image, I show the important components of the in vitro transcription construct, and we'll refer to that several times throughout this presentation as simply IVT. It'll consist of a 5' cap that aids in efficient translation of the transcript to protein, a 5' untranslated region, the open reading frame encoding the antigen, and followed by an untranslated 3' region, and finally the poly A tail. Once this IVT construct is delivered into the cell, it'll produce antigen to elicit an immune response until the mRNA finally degrades. Another mRNA vaccine platform in the bottom here, it encodes the viral machinery which allows this IVT construct to replicate itself once it enters the cell. Now, this method requires much less mRNA to be used for injection because intracellular amplification of the RNA takes place. Important advantages have occurred that have optimized IVT mRNA pharmacology, including capping analogs, capping enzymes, regulatory elements in the untranslated region, length of the poly A tail, modified nucleosides, and codon optimization. So let's take a look at some of the key research areas that have allowed these platforms to be utilized to produce the important vaccines in use today for COVID-19. If we think about the steps that have to happen once the mRNA is injected into our body to elicit an immune response, there are 3 areas of focus. The first, as I described on the previous slide, is to optimize the mRNA sequence such that the rate of translation is high and its half-life in the cell is increased. Secondly, is how the IVT mRNA is purified. It's important that it's free from the double-stranded DNA template as well as transcription components. The IVT mRNA reaction requires several enzymatic steps, and really purification must happen post-reaction. And that's used, typically that's done through HPLC, but some other methods are also employed to purify the mRNA vaccine. The final area of research has been in the area of mRNA delivery into cells. Many areas have been explored here, including nanoparticle lipids, polymers, and peptides. These molecules improve mRNA stability, help reduce toxicity, and facilitate mRNA translocation into the cytoplasm. So let's take a brief look at the modern mRNA vaccine production workflow. What makes mRNA vaccine platforms so attractive is that they enable short development times and include relatively inexpensive manufacturing steps. And these steps are also cell-free, which makes them safer, and sequence independent, meaning the manufacturing steps stay consistent. This is in contrast to some of the traditional vaccine platforms that require changing steps depending on the vaccine. As I mentioned already, we're going to go through kind of a high-level process here, and we've identified four different areas. So in preparation for mRNA production, we first start out by isolating the viral nucleic acid from the host, sequencing the genome, working to choose the antigen that we want to express, and then finally making the expression IVT construct. Once we have that, then we go into mRNA production using in vitro transcription, which starts by linearizing the template DNA, going through the IVT reaction and adding in that template, the dNTPs, and the RNA polymerase to get the mRNA transcript. As I talked about before, we then need to purify. So you'll remove the IVT reaction components, the double-stranded DNA, enzymes, partial transcripts. And this is where, like I said, HPLC can help us along with some other strategies. And finally, we get to the actual formulation step where you add any of the adjuvants that are necessary. You identify and finalize the delivery method formulation, and you add any components that are needed to improve storage and stability. Now, because we're talking about nucleic acids, UV-Vis spectroscopy is a useful tool for QA and QC. And it's useful in a number of reasons. So the firstly is that it doesn't really require any setup or any assays to make your UV-Vis measurements. You can simply measure the nucleic acid natively. And that's because the aromatic rings within the nucleotides absorb UV light. In that graph on the right-hand side, we see the four different bases absorbing light at different wavelengths across the nucleic acids, across the UV-Vis spectrum. Combining those all together into a longer double-stranded DNA or RNA chain yields the result on the bottom there. So in that orange, the orange line is showing, in this case, an RNA sample that the spectral is really a result of all of the individual absorbance patterns of the individual bases. And so using the Beer-Lambert equation, we can take a direct measurement at 260 nanometers and calculate a nucleic acid concentration. Given the equation here, we see that we only need to know the path length that the measurement was made at, as well as the extinction coefficient or the factor, and the absorbance of 260 nanometers, and we can solve for the analyte concentration or the nucleic acid concentration. Different molecules absorb different amounts of light, and that's where that factor comes in. And we can see that the factor changes depending on the molecule of interest, whether that's double-stranded DNA, single-stranded DNA, or RNA. What you'll see here is that we're really focusing on a single wavelength with the Beer-Lambert equation, 260 nanometers. But by eye, you can see that there's much more data here. And in the last 5 or 6 years, the usage of the full spectrum has become more popular, as well as the usage of mathematics called chemometrics to help identify contaminants within the sample. And what this allows us to do is to not only calculate the nucleic acid concentration, but also identify the presence of contaminants and provide a more clear understanding of sample purity. And we'll talk about those software advances a little bit later. So the NanoDrop 8 is helpful in several places throughout the mRNA production workflow. On the left-hand side there, I've put in a little inset image reminding us where we're talking about within this overall process. And it's kind of in between or spans two different boxes here for the preparation of the mRNA production and then the actual mRNA production and in vitro transcription. So as we talked about, one of the first steps is extracting the nucleic acid from the virus and preparing it for sequencing. After you go through extraction, it's important to do a quality control step for your nucleic acid. And this is where you want to measure the concentration of your sample as well as the purity. Then you can go in and sequence the genome, select the vaccine target based on the sequence, and then grow up your plasmid DNA template, grow it into production, and purify it. It's that purified plasmid DNA that you also need to quality control. So you want to look for the sample concentration as well as purity. Finally, you go into the transcription of the template DNA to mRNA and subsequent mRNA purification, pardon me. And then finally, you have that last quality control step, determining your mRNA concentration using the NanoDrop Eight instrument. So now that we have an idea of kind of how the whole mRNA vaccine process works and how UV-Vis technology and the NanoDrop Eight fit in here, we can take a deeper look at the NanoDrop Eight and why it's a useful tool in this whole workflow. So the NanoDrop Eight is really built around the ideas of enabling 8 sample quantity assessment, quality assessment, and improving measurement speed. In the sample quantity assessment arena, the NanoDrop 8 has increased detection limits over the previous NanoDrop 8000. It's now able to measure up to 10,000 nanograms per microliter of double-stranded DNA, or 145 mg per mil of IgG, and it has an expanded wavelength range from 190 to 850 nanometers, same as our NanoDrop 1 spectrophotometer, and this really is helpful for identifying sample purity. Talking about sample quality and sample purity, as I talked about briefly with the usage of full spectral data and chemometrics, we have our Thermo Scientific Aclaro sample intelligence technology that's able to detect chemical contaminants as well as differentiate between DNA and RNA. Not only does it detect the contaminants, it will also provide corrected sample concentrations so that you use the correct amount the first time. Improving on measurement speed, you can measure 8 samples in about 20 seconds or less. And there's some other improved efficiencies as well with the auto blank and auto measure feature so that you can measure your samples much faster than the outgoing NanoDrop 8000 model. All of this can leads you to be able to measure a 96 well plate in under 6 minutes. We talked about concentration and purity a couple of different times. I made reference to our Aclaro software, and this is really how the Aclaro software works. So the applications for the RNA and double-stranded DNA applications, as well as protein A280, are built with libraries of spectra. And so what we can do is we've trained our chemometric algorithms to identify when a sample is contaminated with protein, phenol, guanidine isothiocyanate, or mammalian DNA when measuring RNA. When measuring double-stranded DNA, our algorithms can identify the presence of protein, phenol, guanidine HCl, or mammalian RNA. When measuring protein, our software can detect DNA. On the right-hand side are 2 screenshots of the software and what you would see as a user measuring your sample. In the top, the software has identified a protein contaminant. It shows you in green the original spectrum, and then it will also show you in purple or pink the corrected spectrum, and then in orange, the sample contaminant spectrum. It's able to do this because, like I said, we've built a software, a library, if you will, of spectra that we know has different concentrations of contaminants, and then the software, when you measure your unknown sample, basically it makes a prediction and it compares the sample that you've just measured with our library. And so it can tell you with very high accuracy that your original concentration was measured to be about 400 nanograms per microliter. But in fact, this sample looks like it's contaminated, and it's contaminated with protein. And so your corrected concentration is more like 275 nanograms per microliter. Similarly, on the bottom, we see that this sample was contaminated with phenol. Instead of being a sample of about 400 nanograms per microliter, it's much closer to 160 nanograms per microliter. So what can you do with this information? Well, first, you know what your true concentration is in your sample. And so this is important because in those next steps through the mRNA vaccine production process, you need to ensure that you have proper starting material, a sufficient amount of starting material. If you were to use the original estimate of your sample concentration, then you would be putting in an insufficient amount of template into the reaction. And so you might have reaction problems because you have insufficient template. Depending on what your contaminant is, the contaminant may also inhibit the subsequent reaction, such as if you're using one of the guanidine salts or and some others as well, may actually block the reaction from proceeding. So here you know what the true concentration is, and you have an idea of whether you need to repurify your sample or re-extract your sample so that your future experiments, your future processes go according to plan. Of course, all of this takes place in an environment that is very likely regulated by some type of GXP, right? And so the NanoDrop 8 spectrophotometer also aids in data integrity. As I'm sure many of you are aware, the US FDA 21 CFR Part 11 is really a guidance on data integrity. And in short, right, it employs procedures and controls designed to ensure authenticity, integrity, and confidentiality of electronic records. And so it asks the user, or it enforces the user to ensure that there are validation of systems to ensure consistent intended performance, protection of records to enable accurate and ready retrieval, and limiting system access to authorized individuals. We definitely have validations of systems, so that would be like an IQ/OQ, if you will. But also we have software to enable the bottom two bullet points. So it's a protection of records as well as limiting system access to authorized individuals. And we do that with the Thermo Scientific SciVault software. So this is an optional software. So every NanoDrop 8 does not come with this software, but you can elect to purchase the SciVault software either with your instrument or afterwards. And installing that SciVault software adds a little button on the bottom left-hand side of the NanoDrop 8 home screen. What happens is that authorized users can edit permissions and view audit logs from within the same user interface as the NanoDrop 8, but unauthorized users will not have access to the software and it will be grayed out as we saw before. And it's within this SciVault software that you can allow to put in those checks and those procedures that help you comply with that 21 CFR Part 11 regulation. Really what we are aiming to do here with the NanoDrop 8 and SciVault software is to make compliance less complicated so that you can get back to your production or your research. As I mentioned, it integrates into the NanoDrop 8 software user experience. The user privileges and audit logs are all managed by a single software. And there is a simplified workflow for network installation, as well as software for IT specialists. So what we've done here is we've improved the installation process such that it's easy to control different workstations across a campus, across a company, and then we have the standalone software for the IT specialists or the QA who is monitoring and controlling these instruments, and of course, modern design language in line with our NanoDrop Eight instrument. So one of the things I wanted to kind of walk you through was how easy it is to set up and make measurements on the NanoDrop Eight and be able to input your password and electronically sign the data as you're going through this process. And so the first thing that you would do is that you would blank all eight measurement channels. And this really ensures that you get quality sample measurements. So when making a blank and when making subsequent measurements, you want to ensure that the blank measurement is of the same buffer that's used to suspend your measurements, your samples, I should say. So for example, if you have RNA in water, then you want to blank with water. If you're measuring DNA suspended in TE buffer, you want to be blanking with TE buffer. And as I mentioned, doing this will ensure that your sample results are the best they can be. The next step is the ability to import sample IDs. And so what you can do here is that you can pre-populate a text file with all of your sample IDs. And when you get to this step in the process, you can click on Load Sample ID File, navigate to that file, and go through a couple of screens to identify where in the file those sample IDs lie and import all of those. And this is especially helpful when you have many samples, 25, 100 samples even. So it really just increases measurement efficiency and reduces typing errors so that you don't have to make any corrections or anything like that. It's especially helpful when complying with regulations, so you don't have to electronically sign for any typos that you have to fix. One of the things that we wanted to do also was to ensure that the measurement experience was as efficient as possible. So there are a few things that you can do to set up your software such that once you start making measurements, you never have to go back to the software. You can continue making your measurements. The software will run automatically. And when you're done making measurements, then you can save all of your data. So there's a few, like I said, there's a few ways, a few simple things that you would do to make that happen. In the top left-hand corner is that sample ID input feature where you can load in your sample ID file. And what it will do is then pre-populate the sample name fields from that text file. So all of your sample IDs have been pre-populated. In the bottom left-hand side is a plate map. And so if you have your samples in a 96 well plate, you can place that plate onto the top of the instrument where we have an LED board. The LEDs will light up underneath the columns that are meant to be measured next. The columns can be manually advanced, so you would go from column 1, you can click on the arrows to go to column 2, and as you go through the plate, those sample IDs are automatically pulling in from that file. However, to make it faster, you can turn on auto advance. And what that means is that after you make a measurement, you'll have a few seconds to look at your data, and then the columns will automatically advance to the next one, pre-populating your samples and making sure that you're ready to measure those next set of samples. Talking about automatic measurements, there's an auto measure feature. such that every time you lower the arm, the measurement is automatically initiated. Once you have these three set up, like I said, you simply start making measurements and you don't have to touch the software until you're done making those measurements and you want to save your data and the experiment, because the sample names will pre-populate and fill in for you, and every time you lower the arm, the measurement's initiated. If you are looking to identify DNA from RNA and vice versa, you would simply click on the toggle button for DNA contaminant detection within the RNA application, and that will identify if there is mammalian DNA contaminating your mammalian RNA. We talked briefly, we talked a little bit about the Claro software and how it can identify contaminants and provide you with corrected concentrations. And these are the tools that you use to know that your sample is contaminated so that you can dig in and figure out what's wrong. On the left-hand side, there are two different alerts that you might see. The first is the A in the circle, which stands for a Claro. This is going to tell you that you have a contaminant present in your sample. Simply clicking on that A will bring up that window we saw before, where you can see what your contaminant is, as well as what the true concentration of your sample is. If the software is not able to identify the contaminants and provide you with the corrected concentration, but your purity ratios are still out of tolerance, you'll get the exclamation point in the diamond. And that indicates that a purity ratio is out of tolerance. Clicking on it will give you some advice on what may be causing the problem and suggestions on how to fix that. What you're looking for is that bright green dot on the right-hand side. That indicates that the purity ratios fall within the acceptable ranges, and then the software has not identified any contaminants. What's nice is that after you're done your full plate, you'll have a plate of dots, or a matrix of 96 dots. You can easily pick out what samples are problematic from the samples that have measured properly, and you can make adjustments as necessary. And so the goal here really is just to make it very fast and easy to identify where those problem samples are amongst all of your samples. And again, like I said, clicking on the A brings up the software that helps you prevent failures before they happen. Once you're finished making your measurements, you go to the top right and click on End Experiments. This is where you can edit the experiment name. You can rename it with the details of your sample or your project. If you need help organizing multiple projects, there's a tag feature such that when you go back into the data history, you can search for tags and pull up all of the data for that particular project. Once you're done, you click Save. Then you're prompted for the password as well as the reason for the signing. In this case, we've just finished making measurements. So the reason here is authorship, signifying that I am the one who created these measurements. And finally, after you click OK, you get a notification saying that the data are successfully signed. Of course, if you need to, you can always go back into the history, make modifications. If you need to make a modification, again, you'll be prompted for your digital signature. And if you also are the head of the lab, you're the QA, you can also review and approve data for for future use. Once that data is created, you will likely need to view the audit log at some point. And so again, all within the same software, you can track the data lifecycle. So creation, modification, and even deletion of data is all tracked within our audit logs. There are multiple columns displayed, but you also have the ability to customize these columns, show the data that you need to show, and then save these reports so that that format is always there for you. It's always saved. You can create reports based on the columns that you want, as well as filtering by different things like the username, the source of the data. So depending on where the application is, the date and time ranges. So you can filter and sort to your heart's content, create that report, and then sign the report for your auditing body. And this is all networked across all installations of the NanoDrop Eight software, so you can track events across multiple computers. So to wrap up on how mRNA vaccines are really progressing quickly, right? We've seen a lot of improvement since the 1990s when the first research and the first attempts at mRNA vaccines were put forth. And UV-Vis technology is one of the ways in which the quality of the nucleic acid is really being ensured. And the NanoDrop 8 can help in this regard. However, there are other instruments within the NanoDrop portfolio. Just want to make sure that you are aware of. So there are three main instruments, and they're all suited for really any researcher or scientist's needs. Our NanoDrop 1 is a single channel instrument that does provide full spectral analysis. You can control the instrument either using that built-in LCD display with a touch panel or by connecting the instrument to a PC. When connected to a PC, there is optional 21 CFR Part 11 software. So you can comply with either 8 samples at a time or with 1 sample at a time, depending on the instrument that you want to choose. Our NanoDrop 1 has the widest detection range, measuring up to 27,500 nanograms per microliter of double-stranded DNA, or 400 mg per mil of IgG. Much like our NanoDrop 8, the NanoDrop 1 has contaminant identification as well as concentration correction. So both instruments are built with the same Aclaro algorithms and can identify and detect the same chemical contaminants. The NanoDrop Lite is a basic instrument. It measures only 2 wavelengths, 260 and 280 nanometers, uses a keypad control, and it does have a more limited detection range of up to 1,500 nanograms per microliter of double-stranded DNA. And this can be used as a backup instrument, or other labs like to have multiple NanoDrop instruments, and this is an alternative or a secondary instrument that you might need. We do have a lot of NanoDrop resources, and some are going to be included with this webinar. But if you have questions about other NanoDrop applications or technical resources, there's a link there, and I believe you will have access to the slideshow. So you can click on the link in the bottom right hand to take you to the NanoDrop Spectrophotometer Resource Center. A screenshot of that resource center is on the left-hand side. You can use the search bar to look for your application note of interest, or if you're looking for a particular user manual or some other documentation, this is a great resource. I've called out 4 of our more common or more interesting and recent application notes. All of those images are also linked to the PDF. So one of the questions that we get is for performance data on how we can identify and detect DNA in the presence of RNA and vice versa. So there's a nice application note on how to do that, as well as some method-based application notes on how to quantify gold nanoparticles, how to quantify glucose concentration using a hexokinase assay, as well as the quantification of chlorophyll A and B using a custom method. So in addition to nucleic acid assessment, the NanoDrop microvolume spectrophotometers can be used to quantify a wide range of molecules. The last resource that I wanted to share with you is the Thermo Fisher Scientific Pharma resources page. You can click on that link there or simply navigate to thermofisher.com/brighter outcomes. And the goal here was to really identify and showcase all of the different instruments and services that we have for the multiple steps throughout the pharma process, right? So from early research and discovery to development, manufacturing, and then including compliance. And we have a nice brochure that really explains how all of these different instruments can impact and improve, your workflow, all the way from twin screw extrusion, Raman spectroscopy, FTIR, near IR, X-ray, XRD, rheometry, and of course UV vis. So just a couple of useful resources to educate yourself on the NanoDrop products, as well as the other instrumentation from Thermo Fisher Scientific that can help you in your pharma workflows. So with that, I'll be happy to turn it over with questions. If you want to peruse more about the NanoDrop 8, please feel free to visit us at thermofisher.com/NanoDrop 8. Thank you.
Excellent. Thank you for that brilliant presentation, Patrick. It's now time for our Q&A session. And to kick us off, we have the question, are companies already using NanoDrop instruments in mRNA vaccine development and production?
Great, thanks for the question. So obviously we value the privacy of all of our users, but I can confirm that there are several corporations globally that are using our NanoDrop instruments, the NanoDrop 1 for specifically, since it's been in the market for some time now, in mRNA workflows for mRNA virus workflows for production as well as research.
Excellent, thank you. And along the same lines, what new mRNA vaccines are under development currently?
Yeah, so if we take a look at the publicly available information, specifically there's some nice articles from Nature, as well as some others around some medical college groups. There are a number of mRNA vaccines under development, potentially for malaria, hepatitis, HIV, some specific forms of cancers. So I think we're really going to see a explosion of mRNA vaccines in the next 5 to 10 years.
That's great. Thank you, Patrick. We also have an attendee here who's wondering if it was possible to develop mRNA vaccines for something like foot and mouth disease.
Yeah, that's a great question. I think I have not yet seen any publicly available data saying that a company is working towards that. But it would certainly be nice, right? So I know that that's something that is, still affects a lot of people. So we could be hopeful that a vaccine would be available for that.
Excellent. Another attendee here is wondering, what is the sample size that's needed for the NanoDrop instruments?
Yeah, so that's a really good question. The minimum sample that is needed is 1 microliter, and we can measure up to about 2 microliters as well. So 1 to 2 microliters.
Excellent. And I think we've got time for a couple more questions. The first one being, how can I try out the new NanoDrop 8 spectrophotometer?
Oh yeah, that's a good question. So there are different ways you can try out an instrument depending on your region. So those attendees in North America can reach out to our NanoDrop technical support team and customer support team through the email box nanodrop@thermofisher.com. Again, in North America, you go to nanodrop@thermofisher.com, and we do have a free trial program where you can try out a NanoDrop instrument in your lab for about 7 business days. Internationally, if you're in Europe or in in different parts of the world, you can reach out to your local NanoDrop sales distributor to see what type of programs are available. Some will have demo programs, like I described in North America. Others might be able to do a virtual demo. So there's different ways you can kind of try out the instrument or see the instrument for yourself.
Great, thank you for that. We've got another attendee who is wondering what the limit of detection is for DNA and RNA samples using the NanoDrop 1.
Yeah, good question. So when we think of limit of detection, we're talking about kind of the lower end. So what's the most dilute sample that we can measure? So for the NanoDrop 1 pedestal DNA, that is 2 nanograms per microliter. For RNA, that is 1.6 nanograms per microliter. Thanks for the question.
Thank you, Patrick. Another attendee is wondering, do these spectrophotometers have a Peltier device to help keep the sample temperature constant?
Yeah, that's a good question. So the NanoDrop 8, which we primarily talked about today, does not have any sample temperature control. The NanoDrop 1C that has the cuvette port, it has a simple temperature control system where you can set the cuvette temperature to 37°C. Some of our other cuvette-based spectrophotometers, I'm thinking about our Evolution line from Thermo Scientific, does have Peltier devices, but that would not be on a microvolume scale. That would be in a cuvette-based measurement.
Great, thank you for clarifying that one. And I think we've got time for one more question, and that is, can I measure protein concentration using the NanoDrop instrumentation?
Absolutely. So the NanoDrop instruments are full spectrum spectrophotometers. So you can measure protein concentration in 2 different ways. If you have a purified protein, maybe you have a recombinant protein that you have extracted, purified, you can use a direct measurement at 280 nanometers to measure the absorbance that way. That measures the absorbance due to the amino acids tryptophan, tyrosine, and cysteine double bonds. If you have a peptide that does not contain those amino acids, you can measure using our A205 application, which measures, as you might expect, at 205 nanometers, which is indicative of the presence of the peptide backbone. And lastly, if you have something like whole cell lysate that you might be using for a Western blot, you can use our NanoDrop 1 or NanoDrop 8 to measure a colorimetric assay, something like BCA or Bradford. So there are multiple ways to measure protein using a NanoDrop instrument.
Wonderful, thank you, Patrick. Unfortunately, that's all the questions that we have time for today, but thank you for your presentation and answering the questions that we did have time for.
Thank you.
Thank you very much, and thank you to everyone joining us online as well. I can see that there are still questions coming in, which is great, and we will follow up with you after the webinar, but please also feel free to keep emailing us at editor@selectscience.net with any further questions, and we'll follow up with Patrick after the event. If you'd like to share your attendance or provide feedback using social media, please feel free to mention us using @SelectScience or #SelectScienceWebinars. And remember, you can also download your certificate of attendance in the Related Resources tab. If you'd like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in a few days' time. So goodbye, and thank you all once again for joining us.